Have you ever looked at a pair of identical twins and just marveled at the science? It's like nature hit the copy-paste button, creating two distinct individuals from a single blueprint. But how does this incredible biological magic actually happen? We often hear they come from a single egg that splits, but what does that split really look like at a microscopic level? Today, we're going on an extraordinary journey deep into the world of cellular biology. We'll use stunning microscopy footage to witness the precise, mind-boggling moment when one life path diverges into two. This isn't just about twins. It's about understanding the fundamental mechanics of life itself. Get ready to have your mind blown. So let's set the stage. The journey of every human life begins with a single cell, a zygote. This happens when a sperm fertilizes an egg, creating a unique combination of DNA from both parents. This one cell contains the entire genetic blueprint for a person, their hair color, their height, everything. Normally, this zygote begins to divide and multiply, a process called mitosis, growing from 2 cells to 4, then 8, and so on, eventually forming an embryo that develops into a single baby. For most of us, that's where the story goes. It's a beautifully complex but straightforward path from one cell to one person. But here's where the mystery begins. For about 1 in every 250 births, something extraordinary happens. Within the first few days after fertilization, as this tiny cluster of cells, now called a blastocyst, is making its way to the uterus, an unexpected event occurs. The developing embryo doesn't just keep dividing to grow larger. It splits completely in two. This isn't a chaotic break. It's a precise separation. For years, scientists could only theorize about this process. They knew it happened, but the how was a black box. Why does it happen? The exact trigger is still one of biology's great unsolved puzzles. Is it a random fluctuation in cellular signals? A slight environmental change inside the mother's body? Or is there a genetic predisposition that makes an embryo more likely to split? This moment of division is the central problem we're going to solve today, by going where human eyes have rarely gone before. Now for the incredible solution. Thanks to advancements in time-lapse microscopy and IVF technology, we can finally watch this process unfold. Imagine looking through a powerful microscope at a blastocyst just a few days old. It looks like a tiny sphere made of about 100 to 200 cells. There's an outer layer, the trophectoderm, which will become the placenta, and an inner cell mass, which will develop into the baby. We're zooming in on this inner cell mass. Watch closely. On our screen, you can see the cells vibrating with life, dividing and arranging themselves. Then something amazing happens. Instead of staying as a single cohesive clump, the inner cell mass begins to form 2 distinct clusters. It's not a violent tear, but a gentle, organized separation. One group of cells gathers on one side, and another group gathers on the other. It's as if they receive a silent, invisible signal. Divide. Let's break down what's happening with the DNA. Remember, every single cell in this early embryo contains an identical copy of the original DNA from the zygote. As the inner cell mass segregates into 2 groups, each group takes with it a complete, perfect copy of that genetic blueprint. Think of it like a master document on a computer. You don't rip the document in half. You make a perfect digital copy. That's essentially what's happening here. Each of the 2 new cell clusters has the full and complete instructions to build a human. From this point forward, they will develop independently within the same blastocyst. Sometimes they share a single placenta and amniotic sac. Other times they develop their own, depending on how early the split occurred. But genetically, they are identical clones created by nature. This microscopic view changes everything. It's no longer an abstract concept. It's a visible, biological process. We can see that the split is more of an organized separation of the inner cell mass into 2 distinct developmental poles. Each pole then continues on its own developmental trajectory, reading the exact same DNA script. The result? 2 individuals who are, at a genetic level, indistinguishable. They will have the same DNA, the same blood type, and the same physical traits governed by genetics. Of course, environment and life experiences will shape them into unique people, but their starting point is identical. This process reveals the incredible robustness and flexibility of early human development. Life finds a way not just to continue, but to multiply in the most fascinating ways imaginable. Seeing this DNA splitting event up close really puts the wonder of life into perspective. What was once a biological enigma is now a breathtaking visual spectacle. We've journeyed from a simple question about twins to the very heart of cellular division, witnessing the moment one genetic destiny becomes two. This microscopic ballet is a testament to the elegance and precision of nature. It shows that even at the smallest scale, life is full of surprises and extraordinary mechanisms that we are only just beginning to understand. The science behind identical twins isn't just a curiosity. It's a profound lesson in the resilience and adaptability of our own biology. It reminds us that every life, whether singular or twinned, begins with an incredible microscopic miracle. Thank you so much for joining me on this deep dive into the making of twins. If you found this as fascinating as I did, don't forget to hit that like button and subscribe for more journeys into the unseen world of science. And I have to ask, what other biological mysteries would you like us to explore next? Let me know in the comments below. See you in the next video.